Hey everyone, this is Professor Jeter. I'm here in the microbiology lab at Jackson State with Dr. Ashbaugh. We're going to be doing some demos of the labs that primarily pertain to the information covered pre-midterm. So it's going to be a lot of information on just standard protocol in the microbiology lab and common techniques that are used in regular class settings, and then followed by I don't know, 6 stains, something like that, 5 or 6 common microbiology stains. And Dr. Ashbaugh is going to be demonstrating some of those things for you in this video lecture. We're going to primarily be covering some information on aseptic technique, and then we're going to actually show you how to go about using aseptic technique when you perform a simple staining procedure. So just to give you some quick background information before I cut the camera over to Dr. Ashbaugh to do his demo of aseptic technique and simple staining. Aseptic technique is it's a universal method that's used in healthcare settings, laboratory settings, which the goal of is to just completely prevent contamination. Not only contamination of the things that you're working with, but also of yourself and the people that you work around. So there are some protocols that we want to follow within aseptic technique to ensure that we reduce the likelihood of contamination. And if you guys were able to actually be here in a regular semester outside of a global pandemic, you would what you would find out is that this is a technique that literally every time you enter into the lab and you exit the lab, you follow. And I tell my students, as well as Dr. Ashbaugh, when we do this lab, so to speak, with our students kind of on the first week, during the first week of microbiology lab, that it's really just to get them almost kind of just getting this protocol ingrained into their brain that they have to do this when they come and when they leave lab, because they will be working with live organisms. So that being said, we're going to demonstrate some techniques with aseptic technique, and then we're going to actually go through how to do a simple stain and what that would entail. So I'm going to switch the camera over so you can see Dr. Ashbaugh's demo, and we'll both sort of be adding information in and out through the video lecture. So let me switch it. And there it goes.
All right, so the first thing is our workspace needs to be disinfected. There are likely going to be some bugs there, bacteria, fungal spores, various things are ubiquitous. They're everywhere, and we have to try to reduce the frequency of those in our environment. We need to cut them down so that we can prevent contamination. We're not going to get rid of them entirely. That's sterilization, and we'll be talking about that later some. But the best we can do is disinfection. So I'm going to use a bleach-based cleaner, or there's some other agents that are suited for this. I'm going to go ahead and spray down my work area, if I can get it to go. This might be a little bit slow. And just cover that liberally with your spray. Lysol works well. Phenol-based disinfectants are sometimes used. And I'll let that sit for a little bit, and then I'm going to go ahead and wipe that down. Notice I'm wearing gloved hands. I'm wearing gloves on my hands. Okay. And that's something we want to try to implement as well. All right, so I'll let that dry, and I'll have an area here where I can work and be reasonably confident that organisms have been reduced adequately. So that's the first step. So we'll do that. We'll wipe down when we come into the lab. We'll do our work. And then when we finish, we want to make sure and wipe down as well. We want to disinfect. The next step is, since I'm going to be working with an organism, is to go ahead and take my inoculating loop. That's an inoculating loop there. You can see it's got a wire that has a closed loop at the end. This is a wire known as nichrome. It's an alloy of nickel and chromium, and it's one that is fairly resistant to destruction by heat. And then we've got this handle here. Now the handle is metal. I'm going to put this in a Bunsen burner flame. I'll show you how that's done. For the most part, the heat's not going to be conducted back into the handle. If I were to hold it in there extensively, that might happen. So you learn ultimately just how long this takes, and you avoid that sort of thing. But let me go ahead and light the burner. I'm going to turn on the gas. All right. And I'm going to use this striker, and there we are. We're lit. Notice I've got a nice blue flame here. It means the mixture of air coming in is adequate. A nice hot flame. I'm going to go ahead and flame this loop now. Some people will lay the loop, the wire in there perpendicular to the flame. I kind of like to put it in like this so that it's more in line with the long axis of the flame, I guess. And notice how the whole wire is heated to an orange color.
Let me see if I can scoot that in a little bit. I don't know there you go. That may be a little bit better.
Go. I want that entire wire to be heated to orange, all right? Every last bit of it. Now, this is sterile. I've actually sterilized this. There are no living things on there at present. It'll take a while for this to cool off ultimately. Well, it could accumulate some things, but for now it's sterile. All right, and I'm going to take, I'll have to sterilize this again. I'm going to set that aside. Let me show you what I'm going to do with my slide and my organism. This is a microscope slide. I'm going to go ahead and mount an organism on this. The first step is to place a drop of water on it. Since you're not actually here doing this, it's something initially that a lot of students kind of mess up the first time, but ultimately through trial and error, that they get the exact right amount of water on there. We just need a single drop right in the center of that. Usually what I'll do is take my loop and I'll get water either from the tap or I could take it from a squirt bottle. Get a drop of water on the loop itself and I'll place it right in the center of the slide. All right, I'm going to go ahead and flame this loop again.
And it is very much important to note, and Dr. Ashball will probably go into some detail about this too, or remind you, but it is very important to note that when you're making these slides, you want less water than you would imagine. The more water that goes onto the slide, the longer it is going to take for the slide to dry.
Yeah.
And then that's going to increase your time in lab. And so it's better to think of this class. This is microbiology, micro, small, so a little goes a long way here.
The width of that little drop on that slide is probably maybe 0.5 to 0.7 centimeters or so. Not even the width of an aspirin tablet, maybe even half that actually. So the next thing I'm going to do, I have an organism here. This is an actual plate of a gelatin-like medium called agar. That's spelled A-G-A-R. There's some nutrients that have been added to it. This is the standard medium, semi-solid medium that we use for plating organisms. And this one has a culture of Bacillus subtilis. Bacillus, that genus is composed of organisms that are rod-shaped. They're elongated. We'll talk more about that later. And they are gram-positive. We'll talk about what that means. But this is a very, very commonly used laboratory organism. And I'm going to go ahead and acquire a sample of this. So go ahead and flame my loop before I do that. If this is a pure culture, if I go in there with an unflamed loop, I'm liable to introduce organisms that don't belong in there. I'm going to go ahead and I'm going to actually have to set this down on the bench top. And I'm going to take my lid, and I'm just going to open it slightly. Again, this is part of aseptic technique as well. I want to avoid allowing organisms from the air to fall on the surface.
I generally tell my students when we're doing this, I tell them think about it like a clamshell.
Yeah.
Just kind of you're clamshelling the lid, so to speak.
So I've got a very small amount of material on there. That's another thing that through trial and error you learn less is more generally. And I'm now going to just go ahead and put that in my drop. And I'm going to spread this out into an area about the size of, say, a nickel. And I'm going to, this is emulsification. I'm making an emulsion of bacteria and water. I spread that out to make a nice thin smear so that it will dry fairly rapidly. Now, I want to go ahead and air dry this, this what we call a smear. Now I'm going to go ahead and flame my loop again. Of course, now it's fully contaminated. I want to flame that entirely to sterilize the loop, and I'll set that aside. All right. And now then I'm going to allow my smear to air dry. And generally you can just leave it sit there, or if you want to expedite things, you can actually wave some bibulous paper. This is a paper that we use when we soak up excess stain and fluids on slides later. We can use that as a little fan to kind of dry that smear, and it has to be entirely air dried. There can't be any liquid water left there. And you'll see the smear will take on this sort of cloudy look, sort of like water drops on, say, a glass that's come out of a dishwasher. Sometimes you'll see this residual little kind of hazy drop that's on there. I'm going to go ahead and wave that. All right, and it looks like it's pretty well air dried. Pick it up and examine it. There's actually a little bit of water left there.
While Dr. Ashbaugh is drying the slide, I want to just take a second to mention that he's been demonstrating that a lot of the work has been done with an inoculation loop. And there's some terminology that kind of accompanies that, because when Dr. Ashbaugh, I'm going to show you on the plate, you can see, maybe better on this side. Man, I don't know. Maybe you can't see. Right there is where Dr. Ashbaugh obtained his sample with his inoculation loop. When he did that, the sample that goes onto the loop is now referred to as an inoculum. And we use that term because what happens is that when we're culturing bacteria, now obviously all we're doing here is we're trying to create a stain to view the morphology, which refers to the shape or structure of the cells. But when we're doing cultures like the one that we're using in this example of Bacillus subtilis, we would be obtaining an inoculum from another culture to streak onto this one. And we would want to use aseptic technique to the fullest extent, as he mentioned earlier, because we would want this culture to remain pure. We would not want it to become contaminated, because if you can see, we've initialed on here B subtilis. Of course, you're having to look at cursive writing written backwards. Right. But it says B subtilis for, it's an appropriate abbreviation for Bacillus subtilis. The thing about that is that if we're not using aseptic technique with our specimens when we're subculturing them, then they will become contaminated, and then any test that we want to run with those organisms may give us false positives, false negatives, depending on the test that we're running.
And I had a pretty small water drop. It still probably was more than I needed.
And once it does dry, we'll show you the dried emulsion on the slide, so you can kind of see what you would want to see.
Yeah, after it's dried, we call that a smear. A lot of times you'll make a smear and you really won't see much. It's really tough to see. This one's actually pretty thick, and when you're starting out, it's often acceptable to kind of err on the side of a little more organism than you would normally use. The problem with really thick smears is that when you stain that, the organisms, the individual cells are so densely packed on there, they're laying right on top of one another, and it's really challenging to see the cell shape or morphology, as we say, and also how the cells are arranged relative to one another. Are they in chains? Are they in clumps, little random clusters? Are they in pairs? What sort of arrangement do they have? That's very helpful, and it's something that varies considerably between species. So really thick smears reduce your ability to see those things sometimes. All right, so that's pretty much dry. I'm going to go ahead and do the next step in the procedure, and that is actually flaming the slide to heat fix it. Okay, this is called a heat fixation. And I'm going to use some test tube holders, a test tube clamp. All right, you can use a clothespin if you want. But we need something to keep our fingers back away from the slide, because I'm going to wave this through the Bunsen burner flame, right through the top of that flame, and I'm going to do it 3 times. All right. Let's see if we can do this so that you can see it. Well, but before I do that, let me show you the smear. If you can see that, see how what formerly was just, clear glass has this sort of cloudy, that cloudy smear on it. That's actually a very thick smear, but like I say, for a beginner, that's perfectly okay. So I'm going to go ahead and heat fix. This is heat fixation. So the first step was to flame the loop. Second step was obtain an inoculum to place in a drop of water on the slide. Then I made my emulsion. I air dried, and I'm going to heat fix. And I'm going to go 1, 2, 3. All right, now it's heat fixed. What I've done is I've actually made that smear more or less baked onto that slide. I've not done this to kill organisms. That's not enough heat to do that. If I heated it to that extent, I probably would shatter the slide. But instead what I've done is I've made that smear more or less fast to the glass so that it's not going to come off when I apply my liquids, my stain reagents. I've got it heat fixed. All right.
And this is a good time, too, to quickly mention that heat fixation is typically used when we're trying to create what we would refer to as a permanent mount. Whereas if we want to view this slide multiple times, we're able to do that, compared to some techniques we'll talk about in the class where we discuss wet mounting, which won't result in permanent fixation.
So, well, I'm done with my gas, so I'm going to go ahead and turn that off. All right, I don't need that. Now what I'm going to do is I'm going to apply my stain. Now this is a simple stain. A simple stain is any stain where we apply a single stain reagent. All right, there are stains later that will require 2 stains, a primary stain and a counter stain. Those are called differential staining techniques. But here I'm wanting to use just a single stain. And all I'm doing is trying to highlight the organisms so that I can see them. That's my only concern. I'm not going to learn anything about chemical characteristics of the cell walls or anything like that. I just want to be able to make these cells appear more fully, to get me some contrast between them and the background that I'm viewing them against. So my stain, and you can use any stain reagent you want for this. There are a number of them that stain cells directly. We'll talk about other simple stains where we're not staining the cells directly, but staining the background. That's for later. I'm using one that stains the cell directly, and this is called methylene blue. All right, notice the spelling on that, methyl, METHYL. ENE, methylene blue, very common reagent used in simple staining. Another one that we use sometimes is called crystal violet. We'll use that extensively later. That's important in the gram stain procedure. I could use that one now if I wanted to, but I'm going with the methylene blue. Another one is called safranin. All right, another one that's very commonly used. You'll hear about crystal violet and safranin until you'll probably be tired of hearing about them later. But I'm going to use the methylene blue now. What I'm going to do is I'm going to put that directly on my smear. I'm going to flood the smear. All right. I'm going to cover it with methylene blue if I can get my dropper to work properly. There we go. The whole smear completely flooded. And with this technique, there's a little bit of flexibility in terms of how long that's applied, but the rule of thumb is at least 60 seconds. With other techniques, the timing, the duration of application of reagents has to be more precisely determined. But here I'm just going to keep that on there for about 60 seconds, 60 to 90 seconds, somewhere in there is going to be adequate for that. I don't know if you can see what the slide looks like with the smear on it. Let me see if I can angle it down, pull it closer. So, there it is. So there's my slide. This is a little handy little stain pan. Sometimes you can do this directly over a sink if you've got one outfitted with a screen or something in it to do that. But this little stain pan will catch all my reagents. So that's probably close to about a minute that has elapsed. Okay, so that's applying the stain. The next step is going to be rinsing the stain off. And I'm going to use this water bottle here, a little squirt bottle. This is just plain old water that's come out of the tap. And I can go ahead and rinse that slide. Now what I'm going to do is I'm going to put a gentle stream of water at the top of the slide. I'm going to take the slide and angle it down a little bit, hold it at a slant like this. I'm going to go ahead and rinse both sides until the water that's running off, the eluent, is more or less clear. And now the next step is to dry this slide. And this is where my bibulous paper that I was waving around earlier will come back into place. So there's my bibulous paper, a little booklet of bibulous paper. And all I'm going to do is take my slide and I'm going to open the bibulous paper booklet, find some clean pages, and lay that wet, sopping wet slide in there and gently now sort of press down on that. I don't want to push too hard because I'll break the slide and I'll start this all over again. So, and generally I put it in, a couple of different places, find some dry places in there and do that a couple or three times until the water has soaked off of it. Now I've got a finished slide that's ready now for viewing under the microscope. And we'll be talking about microscopy techniques a little bit later. This is ready to go, right? So what I'm going to do is hang on to this slide, and ultimately then we'll be able to actually view it. I'll take you through the steps used to do that, and we'll be able to see organisms on there. All right. So again, this is a really thick smear. A lot of times you won't see the smear this distinctly when it's done. And those are actually sometimes better slides. But for, quick and dirty, just is anything there? Were there organisms growing on that slide? This will enable me to do that. That's principally why we would want to do that. So I'll lay the slide aside. And now, to finish things off, I can go ahead and dispose of my excess reagents and those will go down the drain. And now I've got a work surface that's potentially contaminated So I'll go ahead and flood that with disinfectant. All right, and then I'm going to wipe that down again. And there I have successfully completed one simple stain using methylene blue. Now, another thing about this, remember I mentioned that the stain literally will adhere to the cells. It will stick to the cells, and they will appear dark against a light background. A more specific name for this particular method of staining is, it is a positive stain, right? And we'll get into this a little bit more. Professor Jeter has up on the board here an explanation of some other aspects of this, but so let's recap. I've done a simple stain because I used one stain reagent. It was a stain that stained the cells directly, so we can also refer to that as a positive stain, right? And then this next thing we're going to talk about is the fact that we're using, for that purpose, a basic dye, okay? So I'll let Professor Jeter, we'll have to set the camera back up for this, but we'll have Professor Jeter go into explanation about why we call this a basic dye. So what's up with that? So if I ask you, what have I done? You could say, well, you simple stained. And this particular version of simple staining is a positive stain, and it utilizes a basic dye.
Let's see if when we turn this around, is the board, is it in the correct configuration? Because when I tried to do the other camera, it was backward. Or is it?
Well, it's pretty small.
Oh yeah, there's no way you can see that.
I can't really see it.
I have to try to move the. Let me unplug.
Did you stop the recording?
No, it's still going. I was just going to pull this over. So yeah, Dr. Ashball did a great job on his demonstration of the simple stain. Pretty thorough explanation on why we simple stain. And I apologize because I think that may be backward to a lot of you. Or maybe it'll be configured correctly when you watch the video, and I apologize because I don't know that I can make that any larger. I've drawn the ugliest picture that I could of a cell on the board, but to just kind of build off of what he was saying, with our bacterial cells, some of the features that we really want to pay attention to that you'll learn more about as we progress through staining is that for most bacteria, and especially the ones we use in our lab, they're going to contain a cell wall, which is the black rectangle I've drawn here on the board. If you can't see it, that cell wall contains a component to it known as peptidoglycan. That's kind of a key component to aiding in the identification of bacteria. Inside of that, we would find the cell membrane, which is traditionally just a phospholipid bilayer, as many of you guys have probably heard about in other courses like anatomy and physiology. Although you might see that with some microbes, they may not always have the bilayer morphology. Like some of the archaea may have monolayers, but we're, again, kind of getting off topic there. The point is the things I've drawn up here, which would include DNA, RNA, protein, electron transport chains, that's ETC, here in the cell membrane. These are examples, as well as the phosphate groups that make up the portions of the phospholipid bilayer. These are examples of things in the cells that carry negative charges. So when Dr. Ashball was talking about positive staining and basic staining, this is kind of the key takeaways from basic staining. The basic dyes like methylene blue that he used in this tutorial, and he also mentioned safranin and probably crystal violet, those are examples of dyes that carry positive charges, So they're known as cationic dyes, indicating they are positively charged. Hopefully, you guys all remember what cations are. Those are ions that possess positive charges. For example, like sodium or something like that. If you've had A&P. But so basic dyes, they're cationic with a positive charge. Thus, they bind to the cells directly, since the cells overall exhibit a greater negative charge. And that imparts color on the cell. It imparts the color of the dye onto the cell. So when we look at Dr. Ashbaugh's cells from his smear, you use methylene blue. Mm-hmm. We should hope that the cells are blue when we look at them underneath the microscope.
Yep.
I think that ends this first tutorial, but we have more to come.
All right. Well, one last thing. So in that stain, it's a solution, and it's made of a solute that's been dissolved in some sort of solvent. Sometimes it's alcohol. Sometimes it's just water. The particles of the solvent that give the dye its color are referred to as chromophores. And that word, I'll spell it for you. I'm away from the board. But it's CHROMO. That's chromo. Phore. PHORE. And that literally means color carrier. So the little particles that are suspended in the solvent are chromophores. And those, in the case of these basic dyes, happen to be positively charged, okay? So again, you got positively charged chromophores, or particles if you want to call them that, and they're going to adhere to negatively charged proteinaceous components of the cells. They're going to cling together, lock together. Those cells are going to stand out, and we're going to be able to see them. If we didn't apply the stain, I'll just tell you, if I put an unstained smear up under the microscope, I wouldn't see anything. I might see ghostly outlines of cells, but I couldn't determine much from that. So hopefully that all makes sense. Now we're going to go ahead and stop this video and set up and do a second stain technique. One or the other of us is going to demonstrate that one. We're going to do a negative stain now. All right?
Derek.